Hey guys and girls, it's Miss Lynham and we are starting our 5th video. I know you are so excited to be listening to this one. It's our 5th one in Unit 3, Cell Structure and Function, and I believe we are starting with the cell membrane. So this should be slide 27. Get your stuff ready and let's rock and roll. Okay, you should know a little bit about the cell membrane because we did talk about it in our biochemistry unit. Plus, you had such an amazing foundation your freshman year when you guys had me for biology, so I have no doubt that you remember a little bit about the cell membrane. Okay, so the cell membrane, also known as the plasma membrane, is selectively permeable. So if we address that property, this is a vocabulary term, so you probably want to highlight this. Selectively permeable means choosy. It's going to choose what goes in and what goes out of the cell. So the job of the cell membrane is to maintain homeostasis. Okay, so it's going to maintain homeostasis by regulating what goes in and what goes out. Only some things can go in and some things can go out, but look at what this is found in. This structure is found in all cells. That means prokaryotic and eukaryotic cells, so it's all-inclusive. All organisms that have cells are going to have a cell membrane. And if you just think about it, its job is to maintain homeostasis. All living things, all living cells must be able to maintain homeostasis by being selectively permeable. Okay, so that's the first property of the cell membrane. Okay, if you look at the cell membrane and what it's composed of, the main component, number one component, is a phospholipid bilayer. And if you remember, we visited those phospholipids in the biochemistry chapter, and they are amphipathic. That means one end is hydrophilic, okay, one end is hydrophilic, and one end is hydrophobic. That's what amphipathic means. And if my pen would cooperate, this would make my life so much easier, but each time I try, it just never seems to cooperate. So we're going to go ahead and write with the mouse here. Okay, so hydrophilic and hydrophobic. Having both hydrophilic and hydrophobic sides to it, that's what amphipathic means, and that's what these phospholipids are. So the bilayer means that there's two layers of phospholipids, that one end of the phospholipid is going to be polar and one end is nonpolar, which leads us to the properties hydrophilic and hydrophobic. Remember, if it's hydrophilic, it's water-loving. Polar things are water-loving, so the polar end of the phospholipid is water-loving, and then the nonpolar region is hydrophobic because it fears water. Okay, I did want to also mention the second main component of the cell membrane are proteins, which also display amphipathic properties. So those proteins, because they're going to embed themselves through the membrane, they're also going to have a hydrophobic and a hydrophilic region to them as well, and we'll examine that as we look a little bit further. Okay, so now I wanted to address the name for the cell membrane, and we call this the fluid mosaic model. We refer to the cell membrane, the plasma membrane, as a fluid mosaic structure. If you just take the word fluid, fluid is because it's not a rigid structure. If it were rigid, it wouldn't allow things to go in and out of the cell. The mosaic part is because there are various proteins that are embedded in the bilayer. Okay, so the fluid is the fluid part, that means not rigid, and then the mosaic is going to refer to the various proteins, the mosaic. Okay, now my pen's going to work. I love it. It's like situational cooperation here. So the mosaic is proteins embedded in the bilayer. So fluid is the part that means allow things through, and then proteins create this mosaic structure. The organization of it is due to the fact that we have, if you look at a cell, woohoo, my pen works on half. If you look at the cell, there's going to be water inside the cell and water outside the cell. So the fact that we have intracellular cytosol and we have extracellular fluid is going to drive the makeup of this phospholipid bilayer, which we're going to look at right now. Okay, so before I switch the slide, I did want to bring your attention to this chemistry model over here. We did this in biochemistry, but you can see the P right here in this part. That phosphate group, remember, is what makes the phospholipid polar. But we also have a glycerol right in here. It's a 3-carbon, do you remember this? 3-carbon alcohol. Okay, that's a 3-carbon alcohol. And then we have 2 fatty acid tails. Okay, one here and one here. And can you notice how this one is showing you a kink? What does that tell you about this fatty acid? Hopefully you were able to say unsaturated fatty acid tail, and this one being in a straight line is a saturated fatty acid tail. We're going to talk about the significance of that in just a second when we look at the picture. But our phospholipid basically has a polar phosphate group, a glycerol, which is a 3-carbon alcohol, connected to 2 long fatty acid tails. So typically, this thing is going to be hydrophilic On the head region, this is the head, the phosphate, and then it's going to be hydrophobic, BIC, in the tail region, which is kind of what you see here. That's what they wanted you to label, is phosphate, polar, therefore hydrophilic, and then fatty acid tails, nonpolar, and hydrophobic. Okay, so if we put these in a bilayer, let me just draw really quickly since my pen is cooperating with me. If I put them in a bilayer, I'm going to get fatty acids like this, fatty acid tails in the middle here, phosphate heads facing this way, and then on the bottom part, I'm going to have a row this way with fatty acid tails coming through. So if this was inside the cell, right here, this is in, and this is outside the cell, notice how they would arrange themselves. These phospholipids would arrange themselves so that the heads face the water inside, and then on this row, the heads face the water on the outside. So the middle region right here in the middle is very hydrophobic, okay, scared of water. But then this is hydrophilic facing the inside and outside of the cell. So that's where we get the bilayer aspect of that, which you can see in this next diagram. Okay, so let's go into a little bit more detail. The organization of the cell membrane or a plasma membrane, I tend to use cell membrane, but a lot of the books do still stick with plasma membrane. But the organization is, the membrane is held together by these hydrophobic interactions. So in this middle part right here, this is hydrophobic interactions between the fatty acid tails. That tends to hold the bilayer together. Okay, the polar phosphate heads, like we said, face internal and external environments due to the fact that there's water inside and outside. And then those nonpolar fatty acid tails face the inside. So those hydrophobic interactions is what holds that membrane together. Okay, these little phospholipids, what's kind of cool, is they can actually shift laterally. Like this one might shift with this one, or this one might shift with this one, or this one might shift with this one. They can flip-flop laterally, a lot like if you were at a concert and you were in a big crowd and you were like elbow to people, you might flip positions this way, but it's rare that you're going to flip-flop a position this way. So when we've examined the cell membrane, you wouldn't see this phospholipid flipping with the one across from it. However, flipping laterally side to side is actually quite common with these little phospholipids. All right. Okay, now we're going to talk about fluidity of the plasma membrane because, like we said, it is a fluid mosaic structure. So let's look at the pictures that you see in front of you, and we're going to talk about the difference between the picture, let's call this A, and picture B. Okay, and we're going to address what we covered in biochemistry. We're going to look at these fatty acid tails. So I'm going to draw really quickly right here a phospholipid. Remember, there are two fatty acid tails, and if they're straight, like you see I drew here, straight down, okay, remember those are tails that have carbon to carbon to carbon to carbon, and then they have tons of hydrogens on the outside. This is what the tail that is straight, like you see right here, okay, let's say this one right here, is a straight tail. What those are saturated fatty acid tails. So I'm drawing the saturated fatty acid tail where you see the carbon chain is completely saturated with hydrogens all around the outside. Remember, that's very hydrophobic, and what that does to the cell membrane, the more of these saturated fatty acid tails that you have, like in picture B, then the less fluid, okay, the less fluid the membrane is. So this one happens to be in picture B. This one is much more viscous. It increases the viscosity when you have tons of these saturated fatty acid tails. But in picture A, you can see some kinks going on here. Those have unsaturated fatty acid tails. And the more unsaturated fatty acid tails that you see, the more fluid-like the membrane is. Okay, so we have the ability in certain circumstances to be able to increase or decrease the number of unsaturated fatty acid tails versus saturated fatty acid tails through the phospholipids that are in our bilayer. Just to give you a couple little stats here, which I think is interesting, but the membrane remains fluid as temperature decreases. So if temperature decreases, it remains a fluid structure. until those phospholipids settle into a closely packed arrangement, such as in picture B. Okay, so at that point, then the membrane will start to solidify as temperature decreases. So the more unsaturated fatty acid tails that you have, like in picture A, when the temperature gets cold, it's kind of preventing that membrane from solidifying. The membrane remains fluid at decreased temperatures, like I said, if it's rich in these unsaturated fatty acid tails. So the more kinks it has, when the temperature falls, the less likely it is for that membrane to solidify, which is a benefit depending on the organism. Okay, the other thing I wanted to show you in this picture below is you can see right in here, these are little cholesterol molecules, if you will go ahead and label that. And if you remember from biochemistry, cholesterol is a 4-ring structure, and it is a lipid. Okay, it is definitely a lipid, and it was the unique lipid because it was a steroid, and it lacks fatty acid tails. Okay, it lacks FA tails. There are no tails. Instead, what you have are 4 rings fused together. Okay, this is the cholesterol. This is found in animal cell membranes. Okay, the job of that cholesterol is stability and fluidity. Okay, so it creates some stability and fluidity in the membrane, and it has different effects at different temperatures. Okay, so just a couple little points I wanted to mention here. At an increased temperature, oh, thank you, Avast. At an increased temperature, which is basically body temperature because it's 37 degrees Celsius, at increased temperatures, cholesterol makes the membrane less fluid by restraining the phospholipid movement. So if you notice, they're wedged in between the fatty acid tails, and the more of these that you have in here, it restrains the movement of these phospholipids. So at an increased temperature, it makes the membrane less fluid by restraining that movement. Okay, however, because cholesterol also hinders the close packing of phospholipids, like the more of these you have in there, it hinders the ability for them to be packed so tightly because it's wedged. It lowers the temperature required for the membrane to solidify. It's kind of thought of as a fluidity buffer, which I thought was an interesting description there. Fluidity buffer is what we refer to these cholesterol molecules as, because it resists changes in membrane fluidity that can be caused by changes in temperature. And they've done a lot of research, okay, a ton of research, and a lot of theories and evolution talk about variations in the cell membrane and the lipid composition. It appears that this is an evolutionary trend. One example I just kind of wanted to tell you about, you might want to write a few of these down, but fish that live in cold environments have a higher proportion of unsaturated hydrocarbons, thus enabling membranes to stay fluid whenever they're in colder environments. So fish are one that they have noticed an increase in the unsaturated fatty acid tails. There's another example about in bacteria, in domain bacteria and domain archaea. They thrive at greater than 90 degrees Celsius in the hot springs like at Yellowstone Park, and their membranes prevent excessive fluidity. Okay, there's another one which is winter wheat, which have an increase in unsaturated phospholipids in autumn to keep the membranes from solidifying in winter when the temperatures get really cold. So that's kind of an interesting trend in studying evolution, is the presence of a lot of these unsaturated fatty acid tails, more of these kinks in the cell membranes of organisms when the temperature gets lower, to try to keep them at a fluid structure as opposed to tightly packed like in this picture to the right. So I thought that was kind of an interesting thing. So just to revisit one more time, these are phospholipids, and then this down here is cholesterol, which is wedged in between the fatty acid tails, and it's only found in animal cell membranes. Plants and fungi and protists do not have cholesterol, which is interesting, but it's used for stability and fluidity. Okay, now let's move on to the protein component in the cell membrane. So the second largest part of the cell membrane, and you can tell in the picture, are these big blobs, like the purple-looking things here, 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 and over here. Those big blobs are the protein component. It's another biomolecule group. That's the mosaic, when we call this a fluid mosaic model. There's an assortment of different proteins that are embedded in the bilayer and pretty much determine most of the membrane's specific functions. Some of these are going to serve as channels and let things through passively. Some are going to serve as pumps and let things through actively. There are some that serve as enzymes, as we studied in the biochemistry unit, which are specific for moving certain things through the membrane. Some have a metabolic purpose. Some act as binding sites for something to attach to the membrane. So there's lots of purposes and functions for these proteins. The two main groups that I like to put them into are the integral. Just look at the word integral. Integral sounds like integrated. These are embedded in the phospholipid bilayer. So this one right here that you see that goes all the way through, this would be an integral protein. Okay, that would be an integral protein because it goes all the way through. This one right here would be an integral protein. This one, integral protein. This one on the top that you see over here is sticking from the outside of the cell membrane. This is a peripheral protein. Peripheral means on the outskirts, so it's bound to the surface of the membrane, and it has a different purpose for that. Okay, so the integral proteins penetrate the hydrophobic interior. Majority are used for transmembrane proteins, which span the membrane, and then peripheral are not embedded at all. They are loosely bound to the surface. Sometimes these might even be attached. These peripheral might be attached to an integral protein. You might see them in two places. Okay? All right, next slide. Okay, hang in there. I think we have 3 more slides left in this video because I'm going to do a separate one on transport. So hang in there. Okay, this next slide is showing you different functions for the proteins. It's kind of a cool little picture. Now I'm going to say a lot, so the good thing is you can pause my video to write a few things down. There's really not any room to write it on this, but I'm going to tell you a little bit about each of the pictures that you see here. These are membrane proteins, and you can see like there's an A and it says transport, B, enzymatic activity, C, signal transduction, D, cell-to-cell recognition, E, intracellular joining, and then F is attachment to the cytoskeleton. So these are all different examples of membrane proteins and what they do. So I'm just going to go through each one of them and say a little bit. If you want to pause and write some stuff down, you can do that. So the first one, and we're going to talk a lot about this in the next video that I post, and that's transport. Okay, my little Kylie is saying hi to y'all. So transport, you can see this picture. Let's just call this picture A, and let's call this little one B. So A and B are channels. A is a channel, and then B is a pump. And so some of these proteins serve as channels, which is letting some things through like this, very passively. And we're going to talk about from a high concentration to a low concentration. B picture can also be a transport protein, but this one's going from low to high, and you'll notice the ATP, which means this kind requires energy. So we're going to talk all about these transport channels, proteins that are channels and pumps. So that's one functioning of membrane proteins. Okay, the B example, we mentioned this one last unit with biochemistry, and that's enzymatic activity. Okay, the enzyme can the enzyme has an active site that can stick to the substrate, bind to the substance, and then do its job. So in this case, you can see this little triangle fits, that's the substrate, fits into the enzyme at its active site, and so does this square in that one. So some of these are going to serve as enzymes on the membrane to do their job. Okay, the C, signal transduction. We're going to talk about this at the end of the unit a little bit, but I just wanted to introduce it now because that's what some of these proteins serve as. It's a membrane protein that is a receptor that may have a binding site that fits the shape of a chemical messenger, such as a hormone. Okay, so in this case, let's say this is the signaling molecule. This might be the hormone that you see in the orange, and on this protein, there's a binding site that attaches to that and sends a certain signal into the cell. So it's one way that cells can communicate by having these binding molecules that do that. So that's signal transduction. Okay, in D picture, this is cell-to-cell recognition. This one is kind of a big topic. It's basically the cell's ability to distinguish one type of neighboring cell from another. It's crucial to the functioning of an organism. It's important in sorting cells into tissues and organs, like during the embryonic development process. It's the basis for rejection of foreign cells by the immune system. Cells have the ability to recognize other cells by binding to molecules, which are carbohydrates, on the outside surface of the membrane. So in this picture, what you see is there are some glycoproteins that serve as ID tags. So here's a protein, has the carbohydrate sticking off of it, that serves as an ID tag that are specifically recognized by this membrane protein on another cell. So if you look at it, cell-to-cell recognition, this is one cell membrane that has these glycoproteins that have carbohydrate ID tags that can be recognized by this cell. And so we're going to talk a lot about that later on. It's a very short-lived process of recognizing, whereas in picture E, you're seeing intracellular joining. which is longer lasting. It's where membrane proteins of adjacent cells hook together in various kinds of junctions, such as a gap junction or a tight junction that we mentioned in the last video. So it's a way for them to stick together in that arrangement. Okay, the last picture, letter F, is where microfilaments can bind to membrane proteins. We showed that with the cytoskeleton. So here's your microtubules, or this one I think is a glycoprotein molecule, and that can bind to the receptor protein to help with structural stability as far as the extracellular matrix, the ECM that we mentioned. So when you see this 6 different types of proteins that are embedded or in the membrane, either embedded or sticking on the outside, that serve a critical purpose in the cell membrane's functioning. Okay, so that's a huge component of the cell membrane, of those proteins. Okay, so I think this is the last component. I believe it is. This is carbohydrate part. So there are carbohydrates in the cell membrane that serve as ID tags. Okay, so you can see in this one, there we go. You can see in this picture that you've got the carbohydrates. You can tell they're carbs because they're shaped like hexagons, which are 6-carbon sugars, and so they protrude from, in this case this one comes from the protein, and there are also some that just stick off of the phospholipids, off the phosphate group of this. So this one is called a glycolipid because it's glyco due to the fact that it's sugar, sounds like glycogen, right, or glucose. It's bound to the phosphates on these phospholipids, but then we also have glycoproteins, which are the same. Here's your carbohydrate ID tag attached to the protein molecule, but these are carbohydrates that I'm circling. That is a carbohydrate that is an ID tag that identifies the cell. It enables the cell to recognize each other and foreign cells. We have some literally that identify the cell as, hey, this is a skin cell in such and such's body. This is a liver cell. This is a cardiac muscle cell. So these have specific ID tags. And even on your red blood cells, some of these serve as being A antigens or A ID tags. Some are B ID tags, which can identify whether you have type A blood, type B blood, type AB blood, or type O blood based on the presence or absence of those ID tags that are carbohydrate molecules. Okay, like I said, here's your box where it says some of these might be bonded to lipids, and those are glycolipids, and some are bonded to proteins, which serve as glycoproteins. Okay, so if we put all that together in the last picture, look at this beautiful picture of the cell membrane. It's amazing. You can see, you can pick out like all the different parts. Hopefully you can pick out every little section. We've got phospholipids right here. We've got cholesterol molecules, therefore this is an animal cell membrane. Okay, there's the cholesterol molecule. We've got proteins here. We've got other proteins here. Proteins, all the purple things are the proteins. Some of them serve as channels, some serve as pumps, some are embedded, integral, some are on the surface, okay, which are the peripheral proteins. Some have like attachment sites for the extracellular matrix. Some serve as enzymes, so that you can see all the different components of the cell membrane. So we have carbohydrate ID tags, proteins, phospholipids, and cholesterol. Huge point. Great job.